So I'm really happy to start this first webinar for the ICAN OMICS Lipidomics series, and I would like to thank you for joining it. So today I will be talking about the power of lipidomics in clinical studies. So I will be talking about 40 minutes, and we will then have time for questions in French or English. So also the talk will be recorded just for you to know. So I would like to start by presenting our institute, which is a foundation for innovation in cardiometabolic and nutrition. So one of the main challenges of our institute is to accelerate research and better fight against cardiometabolic diseases. The institute is working to translate scientific discoveries into concrete clinical applications for patients. So to do so, it integrates fundamental, translational, as well as clinical research that relies on state-of-the-art technological platforms and on a network of clinicians and researchers. So we aim at accelerating innovation via biomarker discovery, evaluation of innovative drugs, and new therapies or new medical devices. So to do so, ICAN platforms constitute a comprehensive tool to study cardiometabolic disease through a multi-scale phenotyping, going from clinical with the clinical investigation platform, the biocollection biobanking platform, and the imaging platform. We then have the cellular phenotyping with the ICAN biocell platform with the flow cytometry, human liver biology, and IPS. And finally, the molecular phenotyping with the ICAN OMICS platform with lipidomics and metabolomics. And the challenge is then to make sense of all this data produced by these platforms using our ICAN Intelligence and Omics platform, specialized in data sciences. So today I'm going to focus on the ICAN OMICS lipidomics platform. And I will start by presenting you what a lipid is. So a lipid is a small molecule that is insoluble or very little soluble in water. So the diversity of this class of molecules is really diverse, really high in structure. So we think there is more than 10,000 different lipid species in human plasma. It's also really diverse in polarity, in size, and in concentration. And this is mainly due to the high possibilities of chain length and unsaturations of these lipids, and also the isomery of the unsaturations of that lipids. Lipids play pleiotropic roles in cellular functions. So first of all, they are involved in membrane structure, obviously, by forming bilayers, and they constitute molecular scaffold for protein recruitment, and also they produce the fluidity required for transmembrane protein function. They also play key roles in signaling as chemical messengers for signal transduction, and they also regulate protein function and gene transcription. Finally, they are also involved in bioenergetics by the production of energy via the beta oxidation. They maintain the body temperature, and they're also involved in energy storage. So for all these reasons, lipidomics, which is the science that studies the lipid composition in a given biological matrix, has emerged as a distinct discipline. So it will study the lipid composition of a given matrix, and due to the multiple roles of these lipids, this discipline aims at defining the biochemical mechanisms of lipid-related disease. So lipidomics, together with metabolomics, glycomics, and metalomics, results from the omics cascade on one hand, genomics, transcriptomics, and proteomics, but is also highly sensitive to its environment, such as food intake, pollution, and treatment. So as such, they give a direct readout of the disease phenotype and the pathophysiological changes. So there are excellent indicators of what the system is presently doing. Clinical lipidomics is an emerging field, as you can see here, with the progression of the published studies during the last 5 years. So when will it be relevant to run lipidomics analysis? So as we said before, lipids are involved in many cellular signaling pathways, as you can see here. And all of these pathways can result in many different pathologies. So basically, any disease that will imply dyslipidemia or where lipid metabolism will be altered, then it will be relevant to run these approaches. So in some cases of cardiometabolic disease, such as obesity, insulin resistance, or diabetes, but not only, is also relevant in kidney disease, some cancers, and also neuropathologies. Another reason why lipidomics could be interesting is because it's, as an emerging field, there is still many, it's very attractive because there's still many gaps to be filled. So what do we expect from lipidomic studies? What we expect is identifying new biomarkers of a given pathologies and or specific lipid signatures. And that together with, combined with other omics approaches, identify the metabolic pathways that are involved in the pathology development, and that way improve the treatment and the prevention of that pathology. On the other hand, combined with classical omics data or clinical data, lipidomics aim at improving the risk scores of that disease and thereby improving the prognosis and diagnosis. Altogether, this should help moving towards a more personalized and precision medicine. So when we talk about clinical studies, we usually talk about the matrix, which is blood, which is non-invasively obtained. And the good thing about lipidomics is that blood constitutes multiple compartments that can be used depending on the pathology studied. So one compartment is the lipoproteins. So there are lipid transporters. They are composed of chylomicrons and VLDL, LDL, and HDL. So for historical reasons, as our platform was first created in the team of Anatole Kontouche at the INSERM here in Paris, we have been studying for many years the role of HDL function and composition in various pathologies. And the study of this lipoprotein composition will be relevant in all cardiovascular disease, obviously, but not only, also in all the dyslipidemia-related disease and also infection. Another compartment will be extracellular vesicles. So they are secreted by the tissues and they are involved in intercellular communications. So they are composed of exosomes, microvesicles, and apoptotic bodies. The main advantage of this compartment is that there are disease and tissue specific biomarkers because they are shed by the tissue, and they also carry a pool of a lot of functional biomolecules. So not only lipids, but also proteins, DNA, or RNAs. So as such, they can be relevant in any pathology. We then have the red blood cells, which are non-dividing cells that transport oxygen. And they can be a good model to study disease progression because they present less turnover than lipids in plasma. So they are a good model membrane that can reflect other membrane composition. And they are, for example, used as model for brain synapses. And again, they have been used already in almost all pathologies as a substrate, as a compartment. And the last compartment is the platelets. So platelets are vascular cells involved in primary hemostasis. So they will be, it's going to be relevant to look at lipidomics in these platelets in all thrombosis related disease, obviously, but not only, also in hypercoagulability diseases, like in some cancers. Okay, so I would like now to give you a few examples on how lipidomics has benefits to clinical studies. And I will first start with cardiometabolic disease and then present some examples in cancer and neuropathologies. So the first example is a collaboration that we've run with the team of Joanne Torgman and Isabelle Dugail, looking at identifying a signature of non-alcoholic steatosis, since the challenge of that disease is to be able to diagnose it non-invasively, since up to now it is mainly done by biopsy. So this was done on a cohort of morbidly obese women. And what we identified is that we've seen that in systemic blood, we could identify a signature with high amount of phospholipids, namely phosphatidylethanolamines and phosphatidylglycerols, as well as high levels of ceramides that confers a specific signature for NASH patients. And what we've shown as well is that when combined with the clinical parameters, lipids equally contributed to the NASH signature. So the hypothesis out of that study was that because these NASH patients also presented a gut microbiota dysbiosis, was that the systemic signature that we also observed in portal blood originated from the gut dysbiosis. And to characterize this, the team of Isabelle Dugail and Karen Clément with Brandon Kaiser, we've done a whole characterization and mechanistical study to identify the role of these lipids. So what we've shown is actually that the phosphatidylglycerol were indeed highly correlated with low diversity in the gut microbiota, and that also PG synthesis was highly regulated by the inflammatory response, as you can see here with cytokines. On the other hand, we've shown that phosphatidylglycerols could inhibit adipose tissue lipolysis, and that their livers were highly associated to adiposity. As you can see here, the BMI is highly correlated with the PG levels, which is not the case of other phospholipids. So in conclusion of that study, what we could say is that the PGs are lipids that respond to an inflammation that is regulated by microbiota via endotoxins, and that PGs are also able to regulate adipose tissue homeostasis in obesity. Another example is here how lipidomics approach could improve GWAS studies by adding endophenotypes to conventional endpoint phenotypes. So conventional endpoint phenotypes could be clinical parameters or just the disease diagnosis. And what we aim at doing here in that study, in collaboration with Peter Mortensen in Denmark, is to look at cholesterol-derived metabolites reflecting either cholesterol synthesis, metabolism, or absorption by lipidomic approaches on a preclinical pig model of 500 individuals. And what we've shown is that using these endophenotypes, so desmosterol on one hand, which is cholesterol synthesis intermediate, and the sum of phytosterols on the other hand, we could isolate two GWAS on chromosome 6 and chromosome 13. And when we looked at the power of these GWAS, we could see that desmosterol and the sum of phytosterol gave the higher statistical power from all the other endpoint phenotypes or endophenotypes used. Also, the use of these endophenotypes allowed the identification of one pleiotropic loci on chromosome 13. As you can see here, they're all located the same loci. Associated with all four parameters. So in conclusion of that study, we could improve the statistical power of GWAS studies, even with a really limited amount of individuals. And using endophenotypes, we can also dissect genetic and molecular mechanisms underlying more complex endpoint phenotypes. Another example of a study run by Peter Michler in Baker Institute in Australia that was published in Circulation a few years ago was to look at how the lipidomic profile of the plasma could improve on traditional risk factors for the prediction of cardiovascular events in patients with type 2 diabetes. And here it was a trial of 4,000 patients where he looked at the prediction of cardiovascular events and cardiovascular death. And what they've shown is that adding 7 lipid species to the model, to the conventional model, and 4 lipid species to the conventional model, it could significantly increase the performance of that model. And this was further validated on another cohort of 500 individuals. Last example on the cardiometabolic disease side, it's a study run by the Norwegian team of Lucia in collaboration with Kim Ekrus, who did the lipidomics approach, where here the aim was to use plasma ceramides to predict cardiovascular death in stable and unstable coronary artery disease. So this was done on 3 different cohorts. And they measured plasma ceramides in these cohorts. And what they've shown again here is that measuring ceramides or ceramide ratio, they obtain better odds ratio than using conventional parameters such as LDL particles or ApoB here. And what they've done then is using this ceramide level, they calculated a ceramide score and they stratified their cohorts using this ceramide score. And what they've shown is that stratifying their cohort that way highly correlated with the occurrence of death, while using LDL cholesterol as a stratification factor, the correlation was much less, was worse. And this was true in both stable and unstable coronary artery disease. So we like now to move on neurodegenerative diseases, where quite a lot of studies have also been run, lipidomic studies have also been run. This is a review that is reviewing all the lipids biomarkers that have been identified in 3 different neuropathologies, so in Alzheimer, Parkinson, and multiple sclerosis. So you can see here quite a lot of different lipids that are involved. So you have ceramides, glycerophosphocholines, arachidonic acid, but also glucosylceramides, sphingosine 1-phosphate, sphingomyelin, and cholesterol. An example of, so this is a collaboration we've been doing with Marie-Claude Pitié in ICAN just next door at the Pitié-Salpêtrière. So this was a multi-omics study where, with the aim of predicting the occurrence of Alzheimer's disease in asymptomatic individuals that presented or not an amyloid load. And what we've shown is that when using multi-omics approach, so transcriptomics, lipidomics, and metabolomics, we could differentiate the 2 groups of amyloid positive and negative amyloid load. And that when we combine these approaches, lipidomics, transcriptomics, and metabolomics, we could obtain really high sensitivity and specificity of disease occurrence model. In cancer, there is also now quite a lot of evidence of the benefits of lipidomics in the clinical studies, as this review is here showing all the lipids that have been identified as biomarkers in many different types of cancers. So these are the ones that have been identified in biofluids, but many of them have also been identified in tissue. Most of these lipids biomarkers were identified as diagnostic biomarkers, but not only, some of them were also good candidates for prognosis and predictive factors. And again, an example of a very recently published paper in Nature Communications by a study done by Michal Olkapek in Czech Republic, in collaboration with other laboratories. So he looked at the use of lipidomic profiling in pancreatic cancer. So this is here a big challenge for that cancer, which has the worst prognosis. So early diagnosis is really challenging here. So they used 3 different cohorts, and this study was done in multiple labs, so up to 3 laboratories, and using 4 different lipidomics approaches, complementary approaches. And what they came out with is that using only lipids, they could show that they perform much better in sensitivity and selectivity than using conventional screening factors, such as here the carbohydrate antigen or the cancer-seq factor. And combined with a conventional factor, they obtain the better performance. They have also shown that some of the identified lipids were good candidates for prognosis assessment as well. Okay, so this is now, I would like now to move on the challenges that are associated with lipidomics studies and what you should be careful of when you start designing your cohort. So as an example here, I would like to focus on some confounding factors that are really now identified, such as age, gender, and BMI. And this is a study that was done by Peter Michael in Australia on 10,000 patients with the aim of studying the effect of these factors on the plasma lipidome. So you can see here on this forest plot, so this is all the lipid classes, and all the colors dots correspond to the significantly altered lipids, depending, differing from men to women. So you can see here that most of the lipids are actually altered differentially, whether you are looking at men or women. He then looked at the age effects on plasma lipidome, and again, all the colors dots here correspond to lipid species that are significantly altered per difference of year. And most interestingly is that when you combine the age and the gender, you will see that the age effects will differ from men to women with sometimes opposing effects. And this was also the case of BMI. As you can see here, all the gray and red dots corresponds to significantly altered lipid species per difference of unit of BMI. And again, if you combine it with gender, you can see that the BMI effect is not the same whether you look at men or women. And again, sometimes the effect is also opposing. So as a recommendation, when you design your cohort here, we would really highly recommend that you use really narrow BMI and age distributions, that you use only one gender, man or woman. And if you're using women even, they've also shown in that study, you should also be really careful on the menopausal state, pre or post, since this will also have a high impact on the plasma lipidome. I would like also now to move to the sample collection and storage conditions that you should use, because lipids are really sensitive to degradation via enzymatic reactions, but also non-enzymatic reactions, such as just heat, light, or oxygen. And as just to illustrate that point, this is a study that studied the effect of the anticoagulant that was used and how this affected the plasma lipidome. And as you can see here, using 3 different anticoagulants, you can cluster your cohorts depending on that anticoagulant. We then looked at the effect of processing temperature, of blood processing temperature on plasma lipidome. This was done in collaboration with the ICAN biocollection platform. So we looked at the effect of temperature on the degradation of lipids, and you can see that up to as soon as 4 hours, the samples start to degrade even at 4 degrees. And this is another study that was done on a more longer term, up to 28 days, and that has shown as well that the polyunsaturated lipids are the main affected by degradation. You should then be really careful about the storage of your samples, and this is another study who has compared the storage condition, the storage temperature conditions, and you can see here that from room temperature to minus 200 degrees, the lipidome will basically cluster differently, which means that you have degradation here, and that at minus 80 and minus 200, you start cluster together, which means that you are in good conditions. This is another example of a cohort that we have been working on, and where we have identified a cluster parameter here as well. And when we tried to look at what was the effect, what was the reason of that clustering, we identified a breakdown, a minus 80 breakdown. So we could see it clearly here. So as the recommendations when you process your samples here is to use a unique anticoagulant for the whole study. Use only fasting blood unless you want to look at the effect of the postprandial effect. You should process your sample as quick as possible and on ice. And you should store your sample in -80, but in monitored -80. Try to avoid freeze-thaw cycles, as this will also reactivate the lipases and possibly degrade your sample. Okay, so in the ICAN OMICS platform, we provide customized solutions going from biomarker discovery to pathway analysis and more targeted diagnosis solutions. So for biomarker discovery, we have a profiling method that will screen up to 650 lipid species from all the different classes, the main lipid species found in plasma. We can then target specific pathways depending on the pathway that have been previously identified. The energetic metabolism, the cholesterol metabolism, microbiota metabolism, or phospholipids. And finally, we can have really targeted analysis on a few lipids only using always LCM, SMS, and GCMS approach. So just to detail you here, the kind of lipids and species we can look at. On the energetic metabolism, we can look at energy storage by looking at all the glycerolipids and the cholesterol esters. We can also look at energy production via both beta oxidation products and lipogenesis. On the sphingolipid metabolism side, we can also look at the de novo pathway by looking at the ceramide intermediates, such as dihydroceramide, on the storage, sphingomanase pathway with ceramide, dihydrosphingomanine, and sphingomyelin, also all the glucosyl ceramide pathway, and the catabolic pathway with sphingosine and sphingosine 1-phosphate. On the cholesterol metabolism side, we can look at all the synthesis intermediates of cholesterol by looking at desmosterol, latosterol, or monosterols, and also all the cholesterol-derived metabolites such as bile acids and stanols. On the microbiota side, we have developed with the collaboration of the Metabolomics platform a range of solutions to screen a maximum of molecules produced by the gut. So that goes from the branched-chain amino acids, short-chain fatty acids, trimethylamine oxide, indoles, to the secondary bile acids and stanols. And all that can be screened in plasma, obviously also in feces, tissue, cells, even lipid droplets. Okay, so now just to show you how our platform is integrated in the ICAN workflow, we can be involved straight from the beginning of the study design in collaboration with the clinical investigation platform. So we can help designing the cohort, study the feasibility, and even target the lipids. Sample collection can be done also in collaboration with the biobanking and flow cytometry platforms here at ICAN. And we will then take care of the sample preparation at the platform here, using either tissue homogenization, liquid extraction. We have an automated solution for sample preparation for big cohorts. Sample will then be processed by LCM, SMS approach or GCMS approaches. So these are the three instruments we have. And then with the collaboration with the ICAN IO Data Sciences platform, we can perform all different biostatistic tests that goes from univariate to multivariate analysis. So like the classical PCA, volcano plots, and correlation metrics, but we can also develop customized tools or visualization tools that are adapted to the design of your cohort, of your study. So for example, here we developed this forest plots visualization to look at how lipid classes were affected and lipid chains. We also developed this circular dendrograms visualization tool in collaboration with Anatole Kontouche to look at the specificity and the common signature in different HDL subpopulations. Here you can see the 5 HDL subpopulations, and this can be done in the order of lipid classes, but that what was also proposed in the order of lipid chain length and unsaturation. In the case of multi-omics projects that can be run in collaboration with the platforms of flow cytometry, imaging, and metabolomics, the ICAN Data Sciences will also propose all kinds of tools to better study multi-omics and multi-scale projects. So this is an example here of the MetaMax projects with which aimed at characterizing macrophages from different tissues in different pathologies. We can also run a work analysis, obviously, and we also help at data interpretation via pathway analysis and other customized tools. Okay, so just to summarize what we've been saying during that webinar, that lipidomics can really benefit your lipidomics, your clinical studies by identifying new biomarkers and lipid signatures, and help that way diagnose the disease and help prevent it. But all that has somehow to come back to the patient care. And for that, the challenge now is really to translate these complex approaches to clinical practice. And this is something that is currently done and by the International Lipidomics Society that has launched the Clinical Lipidomics Interest Group last year, which aim at grouping all the lipidomics, the clinical lipidomics platforms in the world. So we are part of that consortium, and the aim is to harmonize our methodologies to better translate it to clinical practice. So the aim is to come out beginning of next year with a published harmonized method that could be then translated. And of course, the next step to accelerate the adaptation of these biomarkers to clinical diagnostic will be to improve technological tools, such as to have them more portable and wearable, but also to use data mining and artificial intelligence to these approaches. Okay, so with that, I would like to thank all our collaborators in the last 10 years, and thank you to everyone in ICAN, so our scientific directors, Anatole Contuch and Philippe Lesnik, Farid Ishu at the Metabolomics platforms, Sora, our technician, and last but not least, Maha Ponaya for all the data analysis. And please do not hesitate to contact me at this address or to visit our website if you need any question. This webinar will also be posted on our website. And I will take any question en français or in English.